eLTER, Integrated European Long-Term Ecosystem, Critical Zone, and Socio-Ecological Research.
So hi, I'm Bess Hardwick, and I'm one of the LIFEPLAN coordinators here in Helsinki. I'm going to tell you a bit about LIFEPLAN and eLTER. I'll go over how LIFEPLAN works in general for the teams and behind the scenes, and tell you what how eLTER fits into LIFEPLAN. I'll tell you about the current status of the project, what we have ready now and what we'll have ready next, and then a little bit about life after LIFEPLAN. LIFEPLAN operates on multiple spatial scales. A part of what we're trying to find out is what is the relevant spatial scale where ecological phenomena should be measured. We have the global scale, where we looks like we're going to have 83 sites on the global scale, and this global scale is alternating between a natural and urban location, pretty close together. And there are lots of eLTER sites on this scale. Then there's so-called national scale in Madagascar and in the Nordic countries, and again there are eLTER sites in the Nordics involved. These sites stay in the same place all the time, so they don't alternate between natural and urban. And the highest density is the hierarchical sampling, which our teams perform themselves in Madagascar and Sweden. But on all these scales, we have the same kinds of 100 by 100 meter plots, where in the center you have a malaise trap, which collects insects, and where the bottle is changed once a week, so you take weekly samples. We have a cyclone sampler that collects fungal spores from the air, and there the teams take 2 24-hour samples every week. Then in the center and in the 4 corners of the plot, we do soil sampling. So we take soil samples, which we will sequence for fungal DNA. And those are taken 8 times a year, 5 compound samples. And again, in the center and the 4 corners, we have 5 cameras, which produce camera trap images. So mostly mammals and birds, things that are warmer than their environment, get caught on these. And we also have audio recording, 5 audio recorders that mostly will record birds and bats. And the 3 first produce physical samples, so they need to be sequenced to produce digital data, which is DNA sequence data. And the other 2 produce digital data straight away. In the field, the teams use the LIFEPLAN app. It's a mobile app, and they scan a QR code on the trap and on the memory card or bottle or vial or whatever they're using. And when they do this scanning, they create a collection activity which records the location and date and time. And this is the metadata which the app sends directly to us here in Helsinki. Physical samples have to be turned into digital data, so they're sequenced at a lab in Canada, and then we get digital data. And the other 2 are already digital data. And the data and the metadata get connected. For the sequence data, it's done by us, but for the camera and audio data, the teams do the connecting themselves. So they have to tell us which week of samples belongs to which collection activity. We collect massive amounts of data. Most of the bulk is audio, which is very large, but we will be collecting in the order of petabytes of data throughout the life of the project, which is 5 years of active sampling. And this is why we have to have this system in order to keep track of everything and to know for each sample where it came from and when it came from. So that's part of what happens behind the scenes. Another thing to note about behind the scenes is that we have 2 coordinators. So we both answer the same email and read the same email, so you don't have to worry about it, but it's just good to know that Deirdre Cardon is in Uppsala, and she's in charge of sort of the hardware side, so equipment and shipping, receiving samples, and she does technical support for fieldwork, for sampling, and also using the app. And myself, I'm in Helsinki, and I'm more on the side of working out contracts and permits. I deal with data after it's digital data, and I do technical support for LIFEPLAN, which is the uploading platform, and I do communications as well. So how does eLTER fit into LIFEPLAN? So this is our sort of global coverage here, global and Nordic sites included in LIFEPLAN. And it's not always 100% clear to me which sites are actually in the eLTER network, but I hope I got it basically right here. So you can see that in our European sampling, eLTER sites are a really major part of our network. And so we have the medium density sampling in the Nordics as planned, but thanks to the eLTER network, we also have higher density sampling in Europe than we thought we would. So we actually have sort of intermediate density of sampling there, which is going to be very useful in analysis as well. So how is sampling going? I believe there are 24 eLTER sites in LIFEPLAN, and 23 of them have started sampling, so that's very good. Here I'm comparing eLTER sites with general global sites overall. And in general, eLTER sites have been some of the first ones to start and have performed consistent sampling, so they really are very much a backbone of the whole network. The issues that eLTER sites have faced are mostly the same issues that all other sites have, but just less. So the issues that have delayed start have been getting contracts done and shipping equipment and consumables and things like that. But again, the issues have been much less difficult than in the general global network. And there are issues that interrupt sampling, so equipment breaks. We've had some supply shortages of consumables and things, so you haven't necessarily been able to get tubes and vials and memory cards as quickly as you needed them. But that hasn't been a big problem, really. Sometimes sites do still have staff shortages, but that's very occasional, and again, much less than in the general global network. Overall, eLTER sites have the best infrastructure in our global network and the best local conditions. And by that, I mean that the sites often have permanent staff. They might even have technicians who are able to do some minor repairs on equipment or all kinds of improvised solutions to technical issues. They have reliable power supply, they have cars, which is not to be underestimated and or taken for granted. And the local conditions are nice because of safety and security issues. There's much less theft or sabotage than in other places. Usually eLTER sites will have their own land where they can do sampling, and that also helps a lot with permissions. Also in European countries, the legal environment is nice and stable, so we know what the laws are and we know what the permit processes are, and that has been much easier than in other places. So what is the current status of the project? Here I have pipelines of all the 5 different sampling methods, how we get from samples to the actual species lists, and species list being what you actually want to get out of this. For Malay samples, they go through sequencing first and also imaging. We also take pictures of the samples, and that step is ongoing all the time. So samples are coming in and they are being sequenced, and quite a big initial batch of samples has been sequenced. So you see the little Malay bottle is already here, almost at the starting to get to the bioinformatics step. So we have some initial bioinformatics actually done for the first samples, but we want to do another round of bioinformatics that will give us even more species. But we will soon probably be able to share with you initial bioinformatics for your sample. So in this first batch, it is mostly actually eLTER sites because they were, again, some of the first to start sampling. What we're working on now is how to share this data with you in a way that you get access to your data, but not to other teams' data. But we hope to announce this very soon. Cyclone samples get sent to Sweden, where we dry them and clean them. And that's been going on. And the first samples are now starting sequencing. So they've arrived at the sequencing lab, and they're going to start doing that. And those are going to go through the same steps. So then we're going to do bioinformatics and get the species lists. Soil samples are probably the furthest behind, partly because permissions for soils are very complicated and a lot of restrictions on where you're allowed to send them and what you're allowed to do with them. But we are freeze drying the samples as they come in, and we are getting very close to being ready to send in the first samples to a lab for grinding and sequencing. And again, after sequencing, there will be the bioinformatics step where we actually give species names to the sequences and find out what species were in there. Camera data was digital to begin with, so we go straight to here. So teams upload the data to us as they collect it every week. And data has been coming in. We have millions of camera images already, and it's coming in all the time. The next step after it's been uploaded is the annotation step. And we are very close to starting the annotation step for camera images. And I'll tell you a bit more about that in the following slide. But after we have collected enough annotation data, then we will be able to train models that will form the actual automated species classification, where they go through all the millions of images and identify the species. For audio data, it's the same thing. Teams have been uploading the data every week as it comes in. And here we are already well underway in the annotation phase. So annotation is going on all the time on Bird Sounds Global. And I'll show you that as well pretty soon. And the same thing again for audio. Once we have enough annotation data, then we can train models to do the automated species classification. So what data do we actually have now? So for Malays, we already have for 22 sites the raw sequencing data with initial bioinformatics. We're about to get another batch of 20. It's downloaded right now. Already 13 of those first 22 are eLTER sites. So you can see there quite concretely how big a part of the first sampling there has been. And we are indeed working on how to share the initial sequencing data with sites if they want it. Of course, you can always just wait for the final species lists if you don't care about the raw sequencing data. But if you have someone on, maybe a bioinformatician who wants to look at it, then that will be made available as soon as we can. For audio data, we already have a selection of data from several sites on Bird Sounds Global. If I can show, can you see my browser?
Yes.
Yes.
Yes, so this is Bird Sounds Global. It's the site where we do the annotation, and you can see that these numbers tell you how many sites are under the blob. So if you zoom in, you see that here we have lots of sites. So these are LIFEPLAN sites from which we have data up there for annotation. So the idea with annotation is that you listen to pre-selected audio clips and identify the birds in there. And this is an open site. Anybody can make an account there and go annotate. But of course, we need the teams especially to contribute to this. And yeah, more sites are going to be added there pretty soon, and we will notify teams once their data comes online there in the next when we next add more data. For image annotation, we are in the final testing phase of this platform, which is called Zooniverse, where we're going to do image annotation. So we actually have two teams testing the site right now, one of which is actually an eLTER site. And then once we're done with that testing, we will start adding teams to Zooniverse in batches, and you'll be notified when your image data is up there and you can go see. But what it looks like is this. You see these 5 image sequences that you can cycle through, and then we see that there's a squirrel. And we need people to tell us what the species are, because there is no existent database of images of all the animals in the world that we could apply to LIFEPLAN data. So then you just tell the scientific name of whatever this squirrel is, and if you can tell some of these things about it, We're going to say if you don't know what it is, and you go to the next one. And there will be a selection of images from your site that can, where every image actually contains an animal. And so you don't have to click through thousands of empty images. There will actually be something to see in every image. Yeah, so the next things to come out will be the annotation of the annotation phase of images, and there will be more audio annotation audio annotation data coming online. And the next raw data to become available will be the imaging data for malaise, so the insect bolt images, where we take pictures of the whole malaise sample. And then at some point we'll get cyclone sample sequencing data, and eventually also soil sample sequencing data. And if you want your data, you will get access to it as soon as possible. So this is again your data. So to finish, I wanted to talk quickly about life after LIFEPLAN. So I know a lot of sites or a lot of eLTER stations are thinking about how to possibly integrate this kind of biodiversity monitoring into your daily lives in the future after LIFEPLAN is over. So LIFEPLAN ends at 2026, the 6-year project. And what you will have to do that from us, so the tools that we are creating, the software, the models that can turn raw digital data, DNA sequence data, images, audio, into species identifications, will be published at the end of the project and will be available for anyone to use. So you will have that like everyone else will. The equipment, the cameras and the cyclone samplers and these things will remain with the site, so we're not going to ask for them back at the end of the project. What condition they will be at that point, I don't know, but I'm sure some of it will still be functional. One thing to think about long term is the cost of DNA sequencing. So in LIFEPLAN, we are bearing mostly the cost of DNA sequencing. And that's something that sites will have to think about if they want to continue with biodiversity monitoring. They do need to find the funding for DNA sequencing in the future. But of course, we do hope that LIFEPLAN becomes a widespread and long-term effort. So the future is up to you. And that's it from me. Thank you very much. The LIFEPLAN obviously is not possible without all the sampling teams all over the world, including the eLTER station, but here are a lot of the sampling teams that are involved everywhere who are actually doing the work. And that's it from me. Thank you.
Terhi, you are muted.
I am muted, yes. Thank you, Bess. I'm just saying that Otso will continue a little bit more on LIFEPLAN, I think.
Okay, thanks very much, Bess, for this pretty comprehensive presentation of LIFEPLAN. So I will just add some remarks on top of what Bess already explained. Let me first come back to what was kind of one of the original motivations why we started this LIFEPLAN project, which is funded through this ERC Synergy program. And that was that we realized, and many others have realized, that biodiversity data are actually really complex. It has all kinds of structures, it's highly dimensional, and so on and so on. So it's really challenging to conduct like statistical analyses that kind of help you to get the most out of those data. And at the same time, those data are really interesting for kind of fundamental statistics development, just because of the reason that they are so high dimensional and complex. And for these reasons, we devised LIFEPLAN as a close collaboration between statistics and biological research. So in addition to this biological research that Bess was mainly talking about, we have this statistical thread of LIFEPLAN, which is led by David Dunson, who is the PI leading the statistical methods development. And one important achievement so far, on top of all of that Bess was explaining, was that we have managed to built a really fantastic team who is conducting research with the data that we collectively collect with the LIFEPLAN project. So here you see not everybody, but a major part of the Finnish, Swedish, and American parts of the LIFEPLAN team. We had an annual meeting this year recently in Finland, partially in Jyvaskyla, where we got to see how the sampling is conducted in practice in that eLTER site, and then we continued in Jyvaskyla. And what is really fantastic with this team is that we have this multidisciplinary expertise in not only in empirical ecology, but also theoretical ecology, citizen science, statistics, bioinformatics, and machine learning. And what LIFEPLAN really is about is to bring this kind of expertise together to try to make the most of the data that we jointly collect in this global project. And in a kind of a nutshell, LIFEPLAN has these 4 components. One is the empirical component that Bess was mainly talking about. Then there is the bioinformatical component, which Bess also mentioned, which is like how to identify the species behind the samples. Then there's the statistical component, which is like what do we learn about ecological processes and their and you know how they behave in the context of environmental change from all of these data. And then a theoretical component, like how do we place the results of this project into more general theoretical frameworks of for example assembly processes. And I will skip the empirical part because Bess explained that already in quite detail, say something about these other components. Now about the bioinformatical component, like we have this huge amount of data. We don't yet have 100 million camera trap images, but we predict that by the end of the program we have something of that order. And if we split the image to 1 minute segments, we may have like 10 of the order of 1 billion of those segments, and maybe we have 10 billion DNA barcoding sequences. So obviously we cannot identify those manually, but we need the statistical and machine learning methods. And if we talk about DNA barcoding, we deal here with our data, we deal with insects and fungi. So they are the species groups that are very poorly known, in the sense that most of the species in these groups are still unknown to science. That we may have something of the order from 1 million to 10 million species in both of these groups, but only a small fraction of them are known to science. And even for a smaller fraction, we have reference data, reference sequence data available. So now a major part of these statistical methods by which we are classifying the species, doing taxonomic placement from the DNA barcoding sequences, a major emphasis is to be able to deal also with those species that are unknown to science. That how do we classify them, how do we cluster them in a reliable way to different levels. That here is a species that belongs to a known genus, but it's an unknown species. Here is an unknown genus that belongs to some known family, and so on and so on. I think we published a pretty nice paper on how to do this with Finnish insects. That was kind of our pilot study for one relatively well-known species group. Currently, we are doing a similar method model for global animals in collaboration with GELF, so the barcode of life data systems. And then the plan is that this classification system that we are developing, which has this kind of a probabilistic nature, that it tells you about the reliability of the classifications, that will be incorporated into the both data systems, so that then it's available for anyone. Well, concerning these bird sounds and camera trap images, Bess already explained the kind of the citizen science approach that we are taking. Maybe the only thing that I would like to add here is that we already have a kind of a proof of concept, so that the data generated by a pilot version of this project resulted in classification models for Finnish birds that were better than the current state of the art. For example, better than BirdNet. There is a paper coming up in Methods in Ecology and Evolution that describes the modeling strategy and the end result of those models. And that also we are opening this possibility of data outside the LIFEPLAN sampling sites to be incorporated in this system, so that we encourage bird and bat researchers who are not necessarily part of the LIFEPLAN sampling program to bring their own data into the system, annotate the data, and then the benefit is for all of us that we have more annotated data. We can build better models for global birds and bats. And then the benefit for those who bring their own data here is that they get those machine learning models that are exactly tailored for their own data, and they can use them for their local projects. Okay, about statistics, I want to say that our main approach is choice species distribution modeling, an approach that we have developed over the past 10 years in our group with many collaborators, which approach is such that it allows you to integrate data on environmental conditions such as land cover data, climatic data, with species community data, possibly bringing also information on species traits and phylogenies in a single kind of an encompassing modeling framework, which then allows you to do prediction and inference at the levels of individual species and species communities, for example, at the levels of community weighted mean traits. One kind of a newish research line that we have in this context at the moment is the aim to integrate network ecology with species distribution modeling and joint species distribution modeling, so that if we have, as often we have also direct information about the species interactions in terms of an ecological network, how can we utilize such information in joint species distribution modeling to better understand what, for example, creates the co-occurrence patterns between the species. And finally, about the theory, we develop theory mainly with agent-based models. So in the past, we have published some papers that explain like the mathematical framework that allows you to both simulate and analyze kind of a general class of agent-based models. And then we have applied these models to develop theoretical community ecology, but also make the link between kind of the theory and data. And as one example of this past research, we used these kind of models to ask that how does habitat fragmentation influence competitive communities by conducting these kind of special explicit simulations. And now one current line of research in this context also deals with this network ecology, that we are conducting these kind of simulations, but having their explicit network structures of those interacting species to generate synthetic data, which we can then analyze with these newly developing statistical models to see whether they are actually able to infer the network properties from the data as we would like. All right, I think we are running out of time, so this is my last slide. What we really try to achieve and what's kind of motivating our research is to improve this predictive aspect of community ecology by integrating empirical, statistical, and theoretical approaches, and we are really happy that this empirical part of LIFEPLAN is going on as nicely it is, because these data that have these hierarchical structure from global to local scales, they are really extremely useful for this general purpose. Okay, thank you very much.
Thank you very much, Otso and Bess. I think this is really interesting and inspiring to see what this kind of network and support from many sites and this kind of community can offer. I think we keep the questions until the end, and we will now hear the experience from the participant eLTER sites. And we will start with Lago Maggiore, and Emanuele Ferradi will be presenting this.
Hi everyone, and now share with you our presentation. And okay, I will present our experience in the LIFEPLAN project here at Lake Maggiore, or Lago Maggiore, if you prefer. And this lake is a subalpine lake, which is located in the northwestern Italy, but actually the watershed is shared by Switzerland and 2 Italian regions. And it's obviously an eLTER site, and our site manager is Michela Rogora, who is working with us at the Water Research Institute, which is located in Verbania, and is the part of the National Research Council of Italy. And we are now about 60 people working here. And we are equipped for field sampling measurements, and we have laboratories for biology, macrobiology, and chemistry. And we also have a guest house if you want to visit for visiting researcher and student. And we study fresh water. Our main focus is the fresh water, but we did it with a lot of approaches. You can see here, from more moleculars to more biology, chemistry. Well, a lot of approaches that very nice to me. And also in the LIFEPLAN team, we have different expertise. And here you can see the 5 people that are the core MAG team. It's our acronym, and they have all different competences, and this is useful for sampling, but I think it's also in the future for the data analysis and the interpretation of the data. But the weekly sampling is quite an effort, and it wouldn't be possible to do that without the help of about 20 colleagues that were helping us, and we are very grateful to them, because otherwise it would be a lot of time for us, only for the 5 of us. And so I want to publicly thank them. And as said, there are 2 types of site. One is a urban site, which is located near our institute. And the other one is a natural site, which is located 1 hour drive from the institute. And obviously all of them are near the Maggiore Lake. And we started in 2021 with the urban site, which is actually a botanical garden located in the city of Verbania. And we have the collaboration of the Ente Giardini Botanici, which is the manager of the site. And they were very helpful, and they help us even in installation of the equipment, stuff like that. And I have to say, this is very easy access to this site, and it's very close to the institute, so it's very simple to do that. And it's also safe for the equipment, because it's close to in the night, and in the days there is always people there, like gardeners, but also tourists. Sometimes they also ask questions sometimes to us. And here you can see in March 2021, we have our installation procedures according to the guidelines of the project. And you can see all the stuff here, the camera trap, the malaise trap, the cyclone sampler, and the audio trap. And these were the first day of the project. And the same day, we also did the first sampling. And that, I have to say, is very straightforward. In just 1 day, you can train a person, and then he or she can do it alone. And it's based on this app LIFEPLAN developed for the project, and based on QR codes and stuff like that. And I have to say, the worst part is to put the microtube here in the cyclone sampler. If you have big hands, it's the worst part, but for the rest, it's very simple and very easy to do it. And so we could train a lot of people in very few time. And here you can see some results. As I said before, the data analysis is still in progress, but for now we have selected some of the photos, the best pictures in the two sites. And here you can see a lot of birds at Villa Taranto in the urban side. And obviously there is a pigeon since we are in a urban environment, but a lot of other stuff. And of course there are also mammals here. Here you can see some of them, but we mostly see them in the dark. But here you can see a nice sequence, the 5 photos that were collected of a squirrel eating a mushroom, reaching for it and then grab it. It's very nice sequence of pictures for me. But as I said, the best photos of mammals are in the dark, in the night. So you can see badgers, foxes, hedgehogs, but also cats, obviously. And these are some of the many photos that we collected. And then in 2022, we moved to the natural site, which is in the park of Ticino and Lago Maggiore. And Ticino River is the outflow river of the Lake Maggiore. And we are also here in collaboration with the management of these parks, this area. And we choose a wood that is composed mainly of Pinus sylvestris. And I have to say this is less accessible, obviously, but we can reach it by car easily. And but you can quite get lost in the wood because it's very the environment's the same, and you have not points that you can follow, but we managed to come out of the woods every time. And here you can see the installation procedures. Here we also constructed a small house for the cyclone shelter to protect it. But the equipment is obviously the same as the urban site. And there are fewer birds with respect to Villa Taranto, but a lot of mammals. Some you can see are similar, others are different. And also here in the night or in early morning, you can see a lot of animals. It's quite nice pictures. And in the overall sampling of the 2 years, we have done 77 weeks of sampling. And the result is more than 4 terabytes of audio trap recordings, more than 80,000 camera trap images, and maladies trap samples, spore and pollen samples, and also soil samples that are collected not all the weeks. So as you can imagine, the effort is great, and so the help of other people is essential. And I think also that the multidisciplinary competence that you have here at the Institute could be useful in the future for data interpretation. And for now, we are happy to be part of the LIFE project team, and we look forward to the other sampling.
Thank you very much, Emanuele. And I think now we will stay in the Pinus sylvestris forest, but jump a little bit northern. So Pauliina Schiestl-Aalto will tell the LIFEPLAN measurements in Hytiälä and in Värriö. Please.
Thanks. Thanks, Terhi. Yeah. So hi everyone. I will now share my screen. Here, I hope you see this.
It's coming. Yes, we see it.
Okay, good. So as Terhi said, I'll tell you about our 2 sites in Finland. These sites and the stations are led by University of Helsinki and the Institute for atmospheric and Earth system research. So one of those is Hyytiälä in southern Finland, and the second is Värriö in northern Finland. And I'm located in Hyytiälä and responsible of this site. And then I've been setting up the measurements in Värriö, but obviously as it's about 1,000 kilometers north from where I'm located, the technical and other stuff at that station are doing this weekly sampling and all the other processing. So these both sites are located at these SMER stations, which are stations for measuring Earth surface atmosphere relations. So at these stations, we have really versatile measurements of ecosystems, atmosphere, and matter and energy fluxes connecting those. And we have a year-round staff at both of the stations taking care of all the measurements that are running. So both of these sites are boreal conifer-dominated forests and mainly Pinus sylvestris. And both of these are Nordic sites in the LIFEPLAN project. So we don't switch between urban and natural, but stay in the same location for the whole 6 years. But even though both sites are conifer dominated and boreal, they are quite different. So the southern site in Hyytiälä, it's of course much warmer, even though it's cold as we are in Finland. In Hyytiälä, we have a site where one corner is in quite a young birch stand, then one corner and the center is in a mid-aged pine stand, and two corners in a spruce stand. But the whole area is managed forest and it's recently thinned. Whereas the Värriö northern side, it's really cold, close to Arctic zone, with mean annual temperature of -0.5 Celsius degrees. It's a conserved forest. It's one of the only forest areas within Europe that has never been managed or felled by people. So there are pines of different ages. There have been forest fires, but then there are also areas where are these trees of many hundreds of years of ages. So both of these sites are also eLTER sites, and I added the games addresses there as well. There are some photos of the sites showing the differences. So there are these left ones from Hyytiälä, so you see the birches and pines, and then the older spruces there. And then there's the much more open and older Värriö site here, the right side. There's also my son helping when we were setting up the measurements. We have also got some really nice pictures of mammals in both of the sites. And also here we have the best photos, I think, or at least most of the photos during dark time. What I could say of our experiences, we are mostly, but by far, everything has gone really, really nicely. The weekly sampling does not take too much time, even though there's really in Hytiälä only 2 of us doing the sampling. But everything goes really, really well, and it's not too much of a work. The issues that we have are mostly related to winter time. So sometimes the instruments are buried in the snow, and sometimes it's so cold that changing these small memory cards is a bit difficult. And then the cyclone sampler doesn't seem to like the freezing and thawing temperatures too much, so we had to stop the cyclone sampling for a couple of months during the winter time. But otherwise it has gone really well. There's this photo where you can, not so well, but anyway, see a lynx going there. So I've been hoping, this is about the experiences, I have been all the time hoping that we would see some big beasts, because we know there are bears and wolves in this area, but we haven't at least yet captured them. And it can be possible also that they are not coming really that close to the station where there's all the time some sounds of pumps and other motors of other instruments as well. But I'm still hoping that during the project see some bear or wolf. About the future and also for now. So now we are really happy to be part of this project. Especially in Hytiälä, we have had very few biodiversity related measurements so far. We have some series of moths and bats and wasps. And well, from Värriö, we have a much longer time series of, for example, nesting birds, but those are really time consuming to collect those series. So we are very happy to be able to develop these biodiversity measurements in both of the places. It's important for the stations, and it's important for eLTER, and we hope to continue the sampling also after the 6 years. And for now, we are also planning a master's thesis work for a master's student about species diversity in soil within the LIFEPLAN area, where we aim to connect the species diversity with some soil properties. Thanks. That was everything I had from our sides.